Okay, so here we go. This is chapter 2, which is concepts in biochemistry. I tried really hard to get the video links to work in this video, but there's just no way I can do it. I've recorded the entire hour-long presentation 4 separate times, thinking that I might have gotten it that time, and it never worked. So this is the reason for the delay, and you're just gonna have to pause the video. There's only 3 videos I want you to watch, and I really need you to pause this video, go watch that video, and then come back, because I'm going to reference things from the videos, and I need you to understand those concepts. So anyway, here we go with chapter 2. All right, so what is biochemistry? Biochemistry is the study of life at the molecular level, which of course means a biomolecule is any molecule that is produced by a living cell or organism. So if biochemistry is the study of life and biomolecules are the study of cells that are alive. It is essential for a dental professional to have a basic understanding of biochemistry, because if you're going to understand the concepts that are presented in nutrition, then you're going to need this. But also, it's important to understand in your special needs classes, and it's important to understand, in perio. I think that this class will give you a greater understanding of how some of those cells work and interact as far as the immune system goes. And when things aren't working properly, typically the dental hygienist is just sort of advantageously positioned to pick up on some of the signs and symptoms of when things aren't working properly. The 3 major areas of biochemistry that we are going to talk about will be the structure, which is the arrangement of atoms in a molecule in order to give it different groups. Metabolism, which is the production and use of energy. We'll probably be referring to it more as the sum total of all of the energy input and energy output. And then bioinformation, which is the transfer of biological information from DNA to RNA, and its whole process of traveling to the ribosome and being transcripted, and the differences between mRNA and tRNA and rRNA and all of that jazz. So some of those fundamentals that you were required to take chemistry in order to get into this course are that you kind of have an understanding of the bonds that atoms are held together with. And the first one is the ionic bond. This is just a really brief review, obviously. So the ionic bond is formed between a positively charged metal ion and a negatively charged non-metal ion. We don't deal with this too terribly a lot in nutrition, but it is important for us when we think about hydroxyapatite, which is the it's what tooth enamel is made of. And so tooth enamel, or hydroxyapatite, is composed of the ionic bonds between calcium, phosphate, and hydroxide. More importantly for nutrition and for the rest of this course are going to be covalent bonds, which are formed when electrons are shared equally between 2 nonmetals. So all of the rest of the bonds. You might have been looking at that last picture. The picture is also on page 34, and you've been like, oh man, that's a lot of bonds. There's a lot going on there, right? Well, in order to categorize and organize our groups of chemical structures and these molecules and how they all work, we break them down into functional groups, which does exactly what it sounds like they do. It breaks them into groups based on their structure, and based on structure is how those molecules function, right? The way they're shaped is how they work. And so we break them into those groups. Then once they're in groups, if they run into chemical reactions, then they can be changed and converted into other functional groups. We can break them down and rebuild them, because essentially everything is just made up of building blocks. So here we talk about the oxidation, reduction reactions. They're important for metabolism because biomolecules can be degraded, so they can be broken down, or they can be synthesized, built back up again, kind of like Legos. People are Legos. Anyway, oxidation is defined as a loss of electrons or an increase of charge, right? Because when we lose an electron, which is negative, then obviously the atom we're left with is a positive one. This is done in a molecule either through a gain of oxygen, which is it has less electrons, or we can lose our hydrogen atoms. And then reduction can be defined as a gain of electrons. It's the opposite. And so therefore it's a decrease in charge. It means it becomes either less negative or positive. And a loss, this is done in a molecule through a loss of oxygen atoms or a gain of hydrogen atoms. And then condensation is when we create a new molecule by forming a bond between two smaller molecules. If you kind of imagine like a glass of, a cold beverage, when it's hot, you know how like the little beads of water begin to form of condensation, and then eventually they like run together and they will fall that way. That's kind of how condensation works. Things kind of collect individually, they end up forming together, and they create a whole new thing. Hydrolysis, which I struggle with this word because all I see is hydrolysis, so I'm going to say hydrolysis a lot. I don't mean that. I mean hydrolysis, but this word kicks my butt. So hydrolysis is a reaction where it breaks down larger molecules into two smaller molecules. It's pretty important to us as humans because this is how we break down our food. Because we need to break down our food in order to get it small enough to be able to absorb nutrients, and we need to be able to absorb those nutrients or we will die, right? So it's pretty important that we have hydrolysis. I see the word hydrolysis because hydro means water and lysis is kind of the Latin word for cut, and so without water we cannot break down our food. All of the nutrients we talk about are broken down and synthesized with water, and that is why water is the most important nutrient. Oxidation and reduction, these aren't things we're going to talk about a lot in this class. It's not something that I'm going to test you on, but I just want to give you all of the tools you need in order to pass your board exams, and this topic has come up in some of the board prep courses and the board prep books. So if you see it, I want to help you with this mnemonic device. It is Leo the lion says grr. And here Leo is going to be lose electrons in oxidation and gain electrons in reduction. It's kind of a harder concept because when I see the word reduction, I think of like breaking apart. But what you really have to think about you're like reducing the number of things. So if I put things together, then maybe I have less things to carry. I'm reducing that. So yeah. All right, so if you look in your book on page 35, you'll see table 2.1, which has the 4 major classes of biomolecules. This table has some words on here that are new to us. So the words polymer and monomer. We're going to be using these words a lot because in order to talk about what carbohydrates do, we have to talk about how they're made and what they're made of, and they're made of monomers, right? So if we break things down into building blocks, right, then the building block itself is the monomer. It is the smallest unit of whatever it is being repeated. So therefore, monomer, mono means one, polymer means many, right? So a polymer is sort of a chain of all of these monomers grouped together. So a carbohydrate, which is just another word for a polysaccharide, poly meaning many saccharides, and therefore its monomer is a monosaccharide. Carbohydrates' main function is as a source of energy. It's how people were meant to create or to absorb and make our energy. It also has a somewhat limited capacity of energy storage, and it helps us with structure. Proteins is the polymer for amino acids, being its monomer. It's made up of amino acids. And the main function for protein is to is for structure, and for biocatalysts or enzymes, which we'll talk about a lot. And then nucleic acids are made up of nucleotides, and their function is that genetic bioinformation transfer and, to a certain extent, energy. They help to create energy. And then lipids don't have a monomer because they are made up of more than one type of thing. So we don't give them this, but we'll talk about what they're made of. And then their main function is both as an energy source, albeit a harder to reach and use energy source, but it is a much more long-lasting energy source, and it is kind of an unlimited source of energy. Well, unlimited storage form anyway, and it helps us with our biological membranes because all of our biological membranes are made up of lipids. Okay, so carbohydrate structure and function. The main function for a carbohydrate is for energy metabolism and storage. That's what it is supposed to do. If we look at figure 2.2, you can see there that plants use that photosynthesis. 2.2, by the way, is under table 2.1 on page 35. So plants use photosynthesis in order to take carbon dioxide and water and energy, and they photosynthesize that. They get the energy from the sun, by the way. They turn that into glucose and oxygen, and we as animals will use respiration, and we degrade that glucose and oxygen, and we turn it back into carbon dioxide, water, and energy. That's the cycle of how energy is made from plants to animals. If you see there at the bottom of that one on the right hand side, that C6H12O6, go ahead and highlight that if you have it, or circle it, or bookmark it somehow, because that's going to be important for us. So carbohydrates are classified as monosaccharides, disaccharides, and polysaccharides. Mono meaning one, di meaning two, and poly meaning many. Down on that page as well, at the bottom, figure 2.3, you can see there that it has a diagram of what those monosaccharides look like. And those monosaccharides are actually broken into two more groups, which are aldoses and ketoses. So there is an aldehyde functional group and a ketone functional group. For now, that doesn't matter. You just understand that if you take an aldehyde functional group and you reduce it some more, you'll get a sugar alcohol. And then just like we talked about, monosaccharides are the monomer, and so if you combine a whole bunch of them together, you'll get the polymer of a polysaccharide. All right, so on the left side here, you can see that we have the image from your book on page 35, figure 2.3. We can see that this is the chemical makeup of glucose, galactose, fructose, and sorbose. And these two at the top, because they start with CHO and CHO, these are the aldehyde functional groups. And then down here, on these two bottom ones, it starts with CH2OH. Those are the ketone functional groups. But I don't want you to memorize that. What I want you to take from this is that they all have this 6-carbon chain. My dog sees a squirrel. And because they are all similarly shaped and they're all similarly structured, we react to them the same way. They do the same things in the body when we break them down. If you turn in your book to page 35, you'll see the left, this is the right image here, but in your book it's on the left. And this is what happens when we have a glycosidic bond. So this little plus sign, right? These two guys, the glucose and glucose, the two whole monosaccharides, are going to bond together over this one oxygen atom. And this bond right here is called the glycosidic bond. when it bonds together, that is called a condensation reaction. Basically, we have, if this was a glass of iced tea, right? Here's the bottom of the glass. This is a fun-shaped glass. If this were, water on the outside of this glass, we would have this dewdrop, not dew, obviously, and they would be forming together. And when they reach together here, then they're going to run down the side of the glass, okay? So condensation reaction makes these two glucose molecules form this glycosidic bond. And when they do that, they're no longer a monosaccharide and a monosaccharide. Suddenly they are a disaccharide because they're two. That's why. In your book, if you flip, you'll see your book also talks about sucrose, which is when instead of glucose on this side, we have a fructose. We still need the U. And together, glucose and fructose will make sucrose. Sucrose is one of the sweetest carbohydrates or disaccharides there is, and it is more widely known as table sugar. And then if we were to get rid of this thing, and we were to have, over here we would have galactose, then we would have together lactose. And lactose is that disaccharide sugar that is found in milk and dairy products that, some people have a hard time breaking down. So again, hydrolysis is the splitting of those large molecules into smaller, more water-soluble ones that can be used by cells. Without water, this whole thing will just completely fall apart. It doesn't happen without water. The biological function of carbohydrates involves energy metabolism and storage, right? We learned about that in the beginning, but here they are again because they're important. All carbohydrates are supposed to do is make energy and store energy. Plants use photosynthesis to make carbohydrates, well, the carbohydrate glucose, right? The C6H12O6. And when plants do that, they use photosynthesis to make the carbohydrate glucose. That is called reduction. They are taking all of those carbons, all of those hydrogens, and all of those oxygens, and they are reducing it down to one glucose molecule. And then animals, when we breathe and we eat the food, obviously, we breathe in with our respiration, right? We breathe in oxygen, and we have H2O, and we break apart that carbohydrate glucose, the C6H12O6, and we give off CO2. And so it's called respiration for us because that's how we breathe. And that is called oxidation reaction, right? So we have reduction and we have oxidation, and that's how we sort of we have energy through eating plants, and plants make the energy for us. The thing at the bottom, it's red because I want you to remember what it is. Moving on to protein. Protein is essential for almost every physiologic function, okay? So when it comes to, making energy, right, we then use protein in order to do things, to use the energy we have gotten from carbohydrates, and we will, use muscle and tissue in the sort of functioning for us. So proteins will provide structure. They help muscles to contract. They help to transport and store substances. They catalyze reaction, which means they get reactions going. They get started. They regulate our metabolism to a certain extent, and they support immune system. They are, again, composed of what kind of building blocks? Yes, amino acids. They are made up of amino acids. And amino acids all form together into something called a polypeptide chain. I think that's just fun to say. Polypeptides are chains of amino acids, and they kind of fold around in on themselves If you look at the bottom of page 37, you'll see the amino acids. They kind of form that polypeptide chain, and then they all like fold in around itself, and they make protein. They make muscles and tissue and whatever else needs to be made. They need enzymes, though. Enzymes will catalyze all of these biochemical reactions. And so while we need enzymes in a lot of other ways to break down, other foods, proteins need enzymes in order to be broken down, but then they also need enzymes to function properly as well. Okay, so here we are talking about enzymes. And at this point, I need you to pause this video. I need you to go watch this YouTube video from Amoeba Sisters. It is fantastic. It really does showcase what an enzyme, what it looks like, how it works, what it does. It's amazing. Okay, so go watch that video right now. I'll wait. Okay, and I'm trusting that you guys all went and watched it, so welcome back. The enzyme that breaks down proteins is going to be called protease, not proteases, protease. Enzymes on them have an active site, which you can see here in this image. Here is where the substrate is going to fit into, right, inside the enzyme. And so the substrate will come in. If it fits and it works there, and things are good, then it will change. The enzyme will actually change that substrate into a product. Okay, catalyzes the reaction, the product is produced, and then the product kind of goes on about its day, and the enzyme is still there. Nothing happens to that enzyme. And so it will wait around until another substrate comes along. It will change the next substrate into the product, and then that will go on about its day. So enzymes, as long as you have them and you keep them in an environment where they are happy, then they continue to be used. But if they're not happy, if maybe the pH level is off, or maybe you have a really high fever, so your temperature is off, and they're no longer in an environment that they can survive in, then yes, of course, they become denatured. And then obviously the substrates no longer fit there and the enzyme no longer works. That's what happens a lot with people who have like absorption issues. Their enzymes, for whatever reason, they are denatured. Either they were made incorrectly and so they never worked properly, or something happened to them where they're just no longer being used. Also, we need to talk about vitamins. So vitamins, which if you kind of break that word down, it says vital amines, right? They're very important. That's why they start with V, very important. And they are often precursors of something called a coenzyme. A coenzyme, of course, helps an enzyme. That just makes sense, right? Coenzymes help enzymes. And so if, let's say for instance that this little substrate was shaped like this, right? And this whole little section here didn't happen. This wasn't here, right? Well, then maybe we would have another coenzyme that is shaped just like that, so that when the enzyme and the substrate fit together, the coenzyme is like, oh, that's a little nook. I can fit in there. It fits, and then magic happens, and the substrate changes to a product, and then it does what it's supposed to do. It's made it in life. And so a lot of the time, coenzymes are made of vitamins. In order to make our coenzymes, we need vitamins. All right, so moving on to nucleic acid structure and function. This starts at the bottom of page 38. And we don't talk too much about nucleic acid structure because there's not a whole lot we can do about it. We get what we get. But as long as you are consuming the proper nutrients, then this sort of function and this, whole thing gets carried out like it's supposed to. If you're not consuming a diet that you should, then more sort of mistakes happen in this process. And so we need to at least understand that it is happening sort of simultaneously to all of our other metabolism sort of things. But there's also a Nutrigenomics, which is an emerging field that's looking at the interaction of nutrition and genes. And the idea is that eventually they will personalize people's diets to, prevent or treat diseases. I'm not a big fan, or I don't really subscribe that the idea is going to work. I think because most people know and understand what they should be eating in order to be, optimally healthy, but they still like to eat foods that are bad for them, because we're human beings and we only get a while to live, so you may as well enjoy the food that you eat. And so, I don't necessarily think that people will eat the diet that they're supposed to eat. But anyway, let's move on. So a nucleic acid is the building block of these nucleotides. They are, nucleic acid is the polymer, and nucleotide is the monomer here. And what they basically do is they transcribe the information that is in DNA, and RNA is what is made there, and then it transfers and stores and sort of retranscribes all of that information over in the ribosome. So basically, messenger RNA is kind of hanging out in the nucleus of the cell, and its job is to sort of copy down the code of DNA, and then it only copies down certain sections at a time based on whatever the environment calls for, and it will transcribe its information. It copies it down, and it does so through these nucleotides. The nucleotide, this isn't in your book. I think this is just super interesting. So a nucleotide, there's 4 different types of nucleotides that are in DNA, and they always come in pairs with one another. So sometimes we'll see it's A and T are together. A and T always go together. A is adenine and T is thymine. These guys will always be here. So if you see an A and it's missing whatever is supposed to be next to it, it's going to be a T. And then here you'll see a C is always paired with a G. We're not going to quiz you on this kind of thing. I just think it's super interesting. So, G stands for guanine and C stands for cytosine. And these are the 4 nucleotides that are in DNA, right? DNA is important here. However, when RNA comes in to transcribe things, it doesn't have a whole lot of thymine hanging out. It can't make thymine. And so what it does is instead of thymine, let me erase that one, it makes uracil. And so it'll have U right there instead of an A. But then when it gets over to the ribosome, if it needs to make thymine, then it will. Excuse me. So what happens with this is that there is messenger RNA. Once the code is, transcribed, then messenger RNA takes that little chunk of polypeptides and it will carry it over to the ribosome, right? Over here is where the ribosome is. And this is where protein synthesis happens. So there's this message of, okay, we need to build muscle in the biceps, right? And so the messenger RNA carries this message, okay, bicep muscle, bicep muscle, bicep muscle, all the way over to the ribosome. And then when it gets here, the ribosome's like a factory, right? And it's got all the little amino acids kind of hanging out where they're supposed to be. And the mRNA then sort of drops it off. It's like, here you go, this is your problem. And tRNA and rRNA work to make that protein. Whatever it was that mRNA dropped off, that's what it's going to make. So sometimes mRNA messes up and it doesn't make it to where it's supposed to go, and sometimes tRNA messes up, and sometimes rRNA messes up, and that's okay because it's just one tiny little cell. But if all of them are messing up, then we can end up having some sort of defect or some sort of problem. So nucleic acids, right? This whole section here is composed of building blocks called nucleotides. These are nucleotides as well, but when they're all grouped together, it looks more like figure 2.12, where it has that sugar in the middle and has a ribose, and then it also, on one side, it has a nitrogenous base. This one is adenine, right? As if this thing here. And then phosphate is kind of hanging out on the other side. And that's how those nucleotides transfer over to where they're supposed to go. All right, so once again, we have hit the point where if we were sitting in a classroom, I would pull up a video and we would watch it together, but we can't do that because I can't see you, and we're probably not listening to this at the same time I'm talking about it. You can't because I haven't posted it yet. So I need you to pause this video and go watch this YouTube. It's made by whoever this is, named Osmosis, and for this section, for this book, for this chapter, you're only going to need to watch the first 7 minutes of that video. If you watch it at 2.0, it'll only take 3.5 minutes, and I will see you when you get back. Seriously, go watch the video. Hi, welcome back. You probably saw a lot about the different structures of lipids and how those structures affect the biological function of the lipid itself, right? There's a lot of different types. And so lipid structure and function, the function itself, is going to be for energy metabolism, right? We can use a lot of the energy, and a big part is in storage. There's actually an unlimited amount of storage of these triglycerides and lipids. It's going to help serve as structural components of biological membranes. So the myelin that we wrap around the synapses and things like that, that's important. We need the lipids in order to protect our biological membranes. It helps to provide insulation and protection, right? I never thought of it that way, but maybe I'm just, preparing for winter with all of my insulation. And then it's going to act as hormones that regulate the body. When it functions properly, it will help regulate these hormones, but sometimes it will produce hormones it's not supposed to, because that's what it thinks it's supposed to do, and it's kind of on autopilot. But then there's too many hormones in the body, so if you have too much lipid and adipose tissue and things like that, then they're going to be producing hormones that you might not necessarily want. And lipids are also going to help serve as vitamins. They help serve as vitamins, but then another really important thing, which we'll talk about in metabolism, is that they help the absorption of vitamins. Lipids help to absorb vitamins because some of our vitamins are fat soluble, which means they're not going to absorb through the blood vessels, because blood has more of an aqueous solution. It's hydrophilic, it's not hydrophobic, right? And so our vitamins need a fat that is there, like A, K, and D, and E, I think, are the vitamins that need to have a lipid in order to attach onto and then be absorbed. And then it can also act as detergents in digestion. So as we saw in the video, fatty acids are a pretty complex thing. They're made up of quite a bit of things happening. And the video broke it down for the saturated fats. It talks about trans-saturated fats and what that means for the humans, and then it talks about monounsaturated fats and polyunsaturated fats, which every time I see like the MUFA and PUFA, I think of like MUFA and PUFA in my head, and that's really not helping me, but I think it's pretty funny. Okay, so the characteristics, the properties, and the reactivity of a fatty acid can be understood in the terms of its structure, right? Which we talked about, the cis, well, we didn't, the video talked about cis isomers and trans isomers, which have to do with the way that the fatty acid is how well is it able to pack into structures and into triglycerides, or is it not able to pack into a triglyceride? And then the triglycerides are the storage form of fatty acids, right? They need, however, that, the gluco word. Do I not have an image of this? Triglycerols are there. They get stored as those. Glycerol. Glycerol is what we needed. So, of course, triglycerol needs a glycerol. So the glycerol needs the 3 fatty acids in order to attach to it, and then it can be stored as adipose tissue for whenever the energy crisis happens and they need that energy. And then lipids can be transported through the bloodstream, but it can't just travel through on its own, right? It has to have the lipoprotein. And a lipoprotein is sort of like this structure where the outside of it is hydrophilic, means it can, well, hydrophilic means likes water, right? But for the purpose of this, that means it can digest, it can be absorbed through water and it can travel through water. But hydrophobic, which is what our triglycerides are and what most lipids are, and cholesterol and things like that, those are hydrophobic, which means that they can't just travel around and do whatever they want, right? They have to follow in that phospholipid sort of structure, and they have to travel around in that lipoprotein. This is in your book on page 43. You can see that structure of a lipoprotein. There's a really cool image there for it. And you can see all those little triglycerides. I don't know, those kind of look like little, not octopus, jellyfish. And then the other thing is they kind of look like sperm, you know? So that's what I'm seeing. Okay, last but not least, this is the last YouTube video I'm going to ask you to watch. This one is made by Khan Academy, which, I mean, every time I watch something from Khan Academy, I'm just blown away at how amazing things are broken down and described and understood. It's going to draw a very similar picture to this thing right here. It talks about, you know, the oxidative phosphorylation, although it doesn't say these words, it just says TCA cycle. It has TCA cycle here. It talks about the acetyl-CoA. It talks about oxygen and how oxygen has to go in, CO2 comes out. You can see that here. Oxygen goes in, CO2, this is water, but oxygen goes in and CO2 comes out in this whole cycle right here, which is why we have to breathe. And so anyway, go watch that video, pause this one. Please, 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 if you want to understand the concepts as we move forward, I need you to stop this one and go watch the other video. All right, so welcome back. We're going to be talking about metabolism, and in this one you'll refer to the figures 2.21, so you can see that ATP source, the catabolic reactions and the anabolic reactions, anabolic reaction there. I actually learned that I was saying those words completely incorrectly when I started prepping to read this sort of lecture out loud. I didn't realize that I was mispronouncing anabolic. It's anabolic, and anabolism is correct, not anabolic. You have to put the emphasis on the second syllable of both catabolism, anabolism, and metabolism. So make sure you put the emphasis on the right syllable, because that's important. So in metabolism, metabolism basically is how cells acquire, transform, store, and use energy, right? That's what it does. But basically, if we break it down into kind of a mathematical equation, it is the sum total of all of the chemical reactions in an organism. So, we have both energy input and energy output, and all of our chemical reactions that are, carried out in that process. And if we figure out, we add up all of the positives and all of the negatives, whatever we get at the end, that's our metabolism. It includes the coordination, the regulation, and energy requirements of all chemical reactions, right? So the energy requirements and all of we just talked about that, the sum total of all of the stuff that's happening. Here we do talk about catabolism, where electrons are released from the metabolic pathways of the glycolysis and the TCA cycle. You guys saw the TCA cycle, which I'm going to attempt this word. It's tricarboxylic acid cycle. Hey, I got it that time. This is the 4th time I've recorded this lecture. So, the first time, man, I slaughtered those words. And I just got to a point where I had to laugh because I couldn't say it. But I'm there now, I guess listening to the 4th version of this lecture is a benefit for you, but it's about a 16-hour process for me. So yeah. Anyway, the macros are hydrolyzed into their simpler building blocks in metabolism. So carbohydrate metabolism starts in your book on page 43. You can see the text there. And the major source of energy for the body comes from the degradation of carbohydrates. Carbohydrates is the main source of energy for human beings. There is aerobic oxidation of glucose, which is, we breathe in oxygen, we breathe out CO2, we use water, we break those things down, right, into glucose. Glucose is also the main sugar that we're going for. Glucose is the one that the brain uses as fuel. And you're going to need that little snippet of information for your special needs class, that glucose is the main, goal of carbohydrate metabolism, and it is the most predominant one, okay, glucose, which for my class, you need to know that glucose is C6H12O6, okay? So the aerobic oxidation of glucose, which produces carbon dioxide, water, and energy, gets captured as ATP. And excessive amounts of dietary glucose are then stored as something called glycogen. And glycogen, which we see the picture on, let me find the picture, which page it's on, is on page 37, so you're going to have to flip back, and we see it there compared to amylopectin, right? Glycogen is the human version of this, okay? So we store energy as glycogen whenever we have a little bit too much glucose, so that we can have a really quick, easy access. That's why it has all those little branches. So if we just need a little bit of glucose, we just break off a little piece of it, right? Break me off a piece of that glycogen. And so we store our extra glucose as glycogen, and we store it in our muscles and our liver and things like that. The amylopectin, that is starch. And starch is how plants store their extra glucose, basically. And so when you see these two together, can you see how we might need a very sudden rush of energy? So we need all those branches, but plants they can't really need a rush of energy. I mean, they might be starving, so they might use some of their amylopectin, but odds are they're not going to have to like run away from anything anytime soon. At least, hopefully in my lifetime, they don't have to start doing that. So they don't need as many branches for theirs. Theirs is a bit more solid state kind of thing. Anyway, let's move back to carbohydrate metabolism on page 43. And here we begin the process of talking about the 3 hormones that are going to help to regulate our carbohydrate metabolism. Everybody knows about insulin, right? Because, you know, diabetics are insulin resistant, or they are very sensitive. If someone is very insulin sensitive, it means that they have a very good use of their insulin, whereas someone who is insulin resistant means that they don't absorb the sugars that are in their cells very easily, and so they have a hard time maybe losing weight, or they might be more prone to getting diabetes. But anyway, insulin happens when you're in that fed state. And if you flip to page 44, you'll see them at the bottom there. So the insulin is there when you're in that fed state. There is plenty of blood glucose floating around in the blood. There's glucose in the blood, and so insulin is done. And so insulin's kind of like the way that your cells need something to be able to absorb the glucose, because cells all have that phospholipid layer, and so glucose can't just walk in because it's hydrophilic, and your cell walls are all hydrophobic. So the glucose can't get in without insulin, okay? And that's why we produce insulin when there's a lot of blood sugar. However, when there isn't enough blood sugar, basically your body thinks you are starving. And so it produces glucagon. And glucagon is going to jumpstart your gluconeogenesis. Gluconeogenesis is a process where your body starts to make glucose from any and all sources, okay? It is like, guys, it's real. Right now, we're going to die. We need to start making glucose because, again, the brain can only take glucose, okay? That's its only source of food. So if you don't have any glucose, and the brain is the most important organ, well, according to your body, and so your body's like, everything else doesn't matter, we need to make glucose, so it jumpstarts, your brain pumps out glucagon, which then jumpstarts the process of gluconeogenesis. This is when your liver will convert that pyruvate. Remember in the video it talks about the pyruvate? In the gluconeogenesis, your liver will convert pyruvate, and it usually will take pyruvate along with lactate and amino acids, and it starts making glucose, okay? You're going to need that for your special needs class when you guys get to diabetics. So we have insulin, which is when we're fed, and we have glucagon for when we are starving. But when we need that quick energy, right, we're going to start using some of our glycogen. We need something to break apart that glycogen quick as heck, and we need epinephrine. Epinephrine is our fight or flight hormone that is secreted when we need immediate energy needs. We need to run away from the lion, okay? That is when we activate our glycogen degradation with epinephrine. This is a good thing if you're running away from lions, but it's not really a good thing if you are sitting at your desk reading something that just freaks you out, right? So epinephrine is a good thing when you're having to fight or flee, but it's not always the best thing for when you're just chilling, okay? And this will affect our stress, which we'll talk about more about some of these kind of hormones and all of that. So just kind of hold on to epinephrine, because I know you guys have already talked about it with your local anesthetic. So we'll get there. Okay, so protein metabolism. When you don't eat enough carbohydrates and your body needs energy, your body will prioritize using proteins for energy over using protein and amino acids for protein synthesis. Okay, so what happens is that the protein, the amino acids, will break off the NH3 that is attached to the polypeptides, and the NH3 is then converted into urea and it's excreted. And then what's left are those amino acids. Those then enter two ways into that TCA cycle. So one, if the amino acids are ketogenic, then they degrade down into acetyl-CoA, and they get converted into ketone bodies, which are soluble forms of lipids that can be then turned around and put into the TCA cycle, right? It's not as advantageous for you to use ketone bodies in order to turn them into lipids and then turn them into fuel that way, because it will mess up our acidity in the body. We'll talk about that some more in later chapters. But what we also need to understand is that only some of our amino acids are ketogenic. The other amino acids that we have are glucogenic, and these guys degrade down into pyruvate and they enter into the TCA cycle just like all of the others, and these guys get converted into glucose, which can then be used as a fuel for the body, right? So we want to try to prioritize the glucogenic amino acids and use those for fuel if we absolutely need to, over trying to use our ketogenic amino acids. What's usually a better balance for the body is just that if you eat enough carbohydrates, you'll turn carbohydrates into glucose first. That's all that the carbohydrates really can do, right? And you'll use your protein sources, you'll use those amino acids in order to synthesize new proteins in the ribosomes, which is kind of their primary, goal there. We don't necessarily want to completely cut carbohydrates out and then be lacking in sources of glucose, and then have to bring in our protein sources, because it's just not as good for you as the other ways. So glucogenic amino acids will degrade down into pyruvate, they enter that TCA cycle, and they get converted into glucose. The other thing that this book wants me to talk to you about is the fact that there's a difference between essential amino acids and non-essential amino acids. And the difference there is that essential amino acids, they have to be eaten in order for your body to get them. Your body cannot make them. And so they are essential. Well, it means that they're essential in your diet, right? They're all of your amino acids, realistically, they're all kind of essential, but these ones are essential in our diet. The non-essential amino acids are things that we can make, so we don't need to eat them in our diet. You'll see at the top of page 45, table 2.6, it has that list there of essential, which are indispensable amino acids, and it has non-essential or dispensable amino acids there as well. I've broken it down further, so I'd like to read it to you. You don't need to know the difference between glucogenic and ketogenic amino acids, but I think it's super fascinating and kind of understanding the way that this whole ketogenic diet and all of that stuff works, and I think it's just fascinating. So I'm going to read for you the ones that are glucogenic only. These ones will degrade down into pyruvate, they'll enter the TCA cycle, and they get converted into glucose. These ones from the left side are arginine and histidine, and then skip down to methionine, and then all the way at the bottom, valine. And then on the right-hand side, it is still alanine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, and serine. So everything that's on that non-essential side, except for the very bottom, that tyrosine, those are glucogenic only. So almost all of the non-essentials are glucogenic only, and then just 4 from the left side are glucogenic only. Then we have amino acids that can do both. Either they can be keto or they can be glucogenic, just depending on what your body needs at the time. So these, I actually highlighted these guys a different color because they're for both. So I have the isoleucine, phenylalanine, tyranine, or threonine, however you want to say that one, tryptophan, and then down at the bottom of the non-essentials is tyrosine. Now your body can make tyrosine from phenylalanine, so it's not necessarily, obviously it's a non-essential. It's on the right side. Okay, so the only 2 amino acids that we don't have already figured out are leucine and lysine. These are only ketogenic amino acids. So that means that if you are consuming leucine and lysine, which you should be because they are essential, then if you are also following a low-carb diet, then you're going to be turning those 2 into ketone bodies, which then get turned into forms of lipid and used as fuel that way. Those guys are not going to be converted into glucose and used by the body that way. Okay, so another major source of energy, and the last one that we're going to be talking about, is lipid metabolism. And this one comes from the catabolism of lipids. That makes sense, right? The energy source that we get from lipids is actually greater than carbohydrates. So carbohydrates and proteins, although your book doesn't mention this, give us 4 kilocalories per gram. But lipids will give us 9 kilocalories per gram. So that sounds awesome. It's more calories, but it's a little bit more complex to break these guys down. They're not really meant to be used this way, so your body sort of prioritizes carbohydrates over these. Just like, if I have to break something down, I'm probably going to go with something that's easier to break down, even if it doesn't give me as much, because I, get a little, I'm good. The other thing is that the lipids are anhydrous, which means without water. And so the energy per gram of lipid is higher than carbohydrates. That's the difference there between why lipids give you so much more energy. The enzyme that breaks down lipids is called lipase, and it hydrolyzes those triglycerides into those free fatty acids and glycerol. Remember how a triglyceride is made up of the 3 fatty acid chains and 1 glycerol molecule? So the fatty acids are degraded 2 carbons at a time, which produce acetyl-CoA, which is awesome. It enters the TCA cycle and it produces CO2. In the degradation of those fatty acids, the electrons are captured by those redox coenzymes, and then those get used to make ATP via that oxidative phosphorylation. And then anything extra, right, as with carbohydrates and proteins, when there are extra lipids, they end up getting stored as triglycerides, and they get stored in adipose tissue. So then when blood glucose levels are low and the glycogen stores are depleted and there's no carbohydrates or proteins around to break down, then the body will start to break down those triglycerides that are stored in that adipose tissue. The hormone epinephrine is the one that will activate that triglyceride hydrolysis, which will then give us the 3 fatty acids to be used in the TCA cycle, and glycerol, which gets broken down into glycogen, which gets broken down into glucose, which gets broken down, or not broken down anymore, that's it. And then it gets used in the brain. However, under conditions of fasting, starvation, untreated diabetes, and very, very low carbohydrate diets, there is an excess of those degradations of the fatty acids, which then produce too much acetyl-CoA. Okay, so it kind of gets bottlenecked. We're breaking down all these triglycerides because for whatever reason, your body's producing the epinephrine. It needs to start breaking down triglycerides left and right. And all of those extra FAs, the fatty acids, that are getting turned into acetyl-CoA mean that there's too much acetyl-CoA in the system and it's not being converted like it should. And then what happens when there's too much acetyl-CoA is that they start to be converted into ketone bodies, right? So too much acetyl-CoA turns into ketones. Too much amino acids of the ketogenic amino acids, those turn into ketones as well. And what happens here is that those ketone bodies will travel around in the bloodstream and they do provide a source of energy. However, it comes at a cost. So ketones are very acidic and they will change the pH of our blood vessels to be more acidic. That's why it's called ketoacidosis, because it changes the pH level, which is actually kind of harmful for people. So you don't want to, ketoacidosis and ketones are meant to be used kind of as a survival technique. It's not meant to be used as a lifestyle, okay? We're going to read more about this when we get to chapter 7. So for now, let's just kind of keep in mind that excessive acetyl-CoA, so if we're breaking down too many triglycerides into acetyl-CoA, then they turn into ketones, which for the purpose of this chapter is not necessarily beneficial, but we'll read more about it later too. Okay, so I want to try to do a little bit more recap of things, not necessarily like a summary of everything we talked about, but maybe just at the end I'll kind of bring up a couple of things. So I just have a couple of questions and we'll either, answer the questions, or we will just talk about the topic. Okay, so what's the difference between a starch and a glycogen, right? This one right here, amylopectin, is a starch, and glycogen is, well, it's a glycogen. So amylopectin is used by plants, and glycogen is used by humans. And you can see here, based on all of these little branches, that this one's a little bit more malleable, while starch is very firm and straight, and this one can't be broken down as easily into quick energy. It is an energy that is stored, but it's not really something we can break down quick. Whereas with glycogen, all these little tiny branches where there's like little 2s and little 3s, all of these can be broken down super quick for really quick energy whenever we need to run away from Leo the lion. Okay, so which of these chemical formulas is the chemical formula for glucose? Good job. If you were thinking it was B, you were right. It is C6H12O6. I never knew how to write in subscript before, which if you push Control and then the little plus sign equals, you'll switch over into subscript, and then you just push Control plus sign equal back to get to it. So you learn something new every day. When I took chemistry, we just wrote on paper. So this is cool. Okay, so which word best describes the biosynthesis of more complex molecules from simple precursors? So what this says is we're going from very simple to a bit more complex, right? Is it catabolism? Is it metabolism, catabolism, anabolism, or hydrolysis? All right, so if you were thinking anabolism, you were right. Anabolism is where we build things up. Catabolism is where we break things down. Metabolism is the sum total of everything. So catabolism, hold on, let me erase this. Get rid of that, get rid of that little dot, get rid of that thing. Okay, so catabolism, we are subtracting. Anabolism, we are adding. If you think of anabolic steroids make bodybuilders huge, right? Anabolism makes bigger. Catabolism breaks things down. Metabolism is the sum total. Hydrolysis is just the use of water to break things apart. So this process isn't the same, right? If it were talking about water, then it might be hydrolysis, but it's not. All right, so that's the end of chapter 2. Make sure you watch those videos. If you need to go back, there is a Word document in this week's lecture where you can just watch the videos if you don't want to click through all of the different slides in order to find the link. And there is a handout. I've given you the key as well. So that one goes over metabolism. And my dog sounds like she's so sick of me recording this. But so the handouts will go over the metabolism. There is a key that goes there as well, so you can get those answers and you can prep for the quiz, which is going to be week 3. I don't have the date on me, but you'll for sure have it.